Well, good afternoon, everybody, and welcome back from a break. I hope that you were able to digest the afternoon early session and have a think about what the Brain Cancer Centre might mean to you, whether you're a researcher, an institute, a member of our community, a patient carer. It hopefully rang true for you in some way, shape or form, and we'd love your feedback. With that in mind, we have our expert panel discussion this afternoon, which will be a fantastic way to dive a little deeper into the conversation around brain cancer. We do have Professor Kate Drummond on the line. She has been good enough to, and I think shows the level of engagement that people like Kate have, good enough to join us from home, having just been to the USA and a little under the weather. I don't know if you want to tell people you've got COVID, Kate, or not, but anyway, we just did. About 500 of us.
Thanks for keeping that a secret there, Sam.
Sorry. I really should have checked that with you.
That's great. Here I am in the leper colony of my house and I'm not allowed out, but I'm really excited to be here.
Fantastic. Thanks, Kate. Thank you for joining us. So this afternoon we have some incredible people here in front of us and obviously Kate on the line. We've got Professor Mark Rosenthal, who's a senior staff specialist in the Department of Medical Oncology at Peter MacCallum, actually runs the neuro-oncology division at Peter MacCallum. I really appreciate his attendance. As I said, Professor Kate Drummond, Director of Neurosurgery at the Royal Melbourne Hospital. Kate, she asked me to put this in as well, recently been invited to join the World Academy of Neurological Surgeons. She didn't ask me to put it in, I'm being silly. And is the only Australian to do so and the only non-American member of the editorial board of the Journal of Neurosurgery. So we're very, very lucky to have Kate. The Associate Professor Joe Nicolazzo, based at Monash University and does some work within our blood-brain barrier group. We have Professor Peter Gibbs, who's a medical oncologist, who can raise your hand, otherwise people here won't know. Medical oncologist at Western Health and the head of personalised oncology here at WEHI. We have Dr. Jim Whittle, who's a clinician scientist, obviously, and we've heard from him earlier on in the afternoon. And we have the Associate Professor Misty Jenkins, currently a laboratory head at the Walter and Eliza Hall Institute and researching brain cancer and immunotherapy with a focus on adult GBM and DIPG. Is that right?
Not limited to that.
Not limited to that, but that's right. I don't want to put Misty in a box at all. There is plenty going on. Where we will start, though, is with a really incredible story, and I want to welcome Jean here today. So, Jean, thank you so much for your involvement. Jean is a person currently working through lived experience with brain cancer, and I won't tell his story for him, but he has described the cancer as a physical assault on his body, and I want, if you don't mind, Jean, to take us through your story and grateful for your time.
Thank you, Sam. Can we just get the next slide? Before I tell you about my journey over the last 55 weeks, and it's been exactly 55 weeks, I want to dedicate everything that has happened and that will happen to Luca and Joshua, my two boys. There's been some dark days, and I'm pretty crap at a lot of things, but one thing I'm good at is being a dad. And I want to be a dad to Luca and Joshua going into the future. And my family and friends have been nothing but stunning. So I'm not going to name them all, but God, there's been a hell of a lot of them, and I thank all of them. Can we go to the next slide? Sincere thanks to everyone at the Alfred who's been involved in my care. Again, I'm not going to go through all of them, but the take-home message from here, this slide is it's a multidisciplinary team. I've seen them all, or they've seen me and put up with me, really. And they have got me here today, and I thank them sincerely. Okay, 55 weeks ago, I was in Canberra visiting the Chief Medical Officer. I got up out of my seat, walked across the other side of the room to shake his hand, did that, walked back to my seat to get my things to leave, and I fished for my chair. And I was with two intensive care doctors, and they said, "Oh, you're having a seizure." What? No, I'm not. I'm fine. I'm just been having these dizzy spells for a couple of years. And I just thought, just another day, move on, take a couple more Nurophens and I'll be fine. By the way, if anyone owns shares in Nurofen Factory, thank you. I've supported you tremendously over the years. This is what happened 55 weeks ago. They found what we lovingly call at home the avocado over here. I'd been having headaches for a long time, greater than 10 years, maybe 15 years. But as my two colleagues said, you can't go back to work until you have an MRI. Okay. And they found this guy. It was squashing my right temporal lobe to the point it killed off my temporal lobe. And when I asked the neurosurgeon, estimate, how big was it? And I was being optimistic, thinking it was, the size of an apricot. And he said, imagine a really large sized avocado. And that's probably about it. And so there's two parts to this avocado, low-grade glioma and high-grade GBM encased within the low-grade stuff. 3 days later, had a craniotomy, which I don't recommend, but if you have to do it. And they removed 90-something% of it. Interestingly for me at least, when they drilled the hole here to get in there, it almost fell in as soon as they started drilling away. When I asked why, they said, well, it's probably because the avocado was rubbing up against it for a long, long time. And I said, well, how long? And they estimated could have been up to a decade. There was such a little amount of bone left. If I had a sizable hit to the side of the head, I would have been in a fair bit of trouble. Anyone, sorry, go back.
Sorry, mate.
I'm probably one of the luckiest people in this room, despite this thing. For those who are interested, my midline had shifted over 20 millimetres. I was told if you have more than 5 millimetres, you're in trouble. And I'd herniated a whole heap of my brain. And again, so much so that it was considered a medical emergency, hence 3 days later, this happened. By the way, that's all fluid. As I said, they got greater than 90% of it during surgery. Sorry, Sam.
Next.
Okay. At that point, a few weeks later, I started the STOOP protocol, radiotherapy, and then temolazide, which was unpleasant. And that went on for over 6 months in total. When I started part 2, I got 2/3 of the way through that, and that's just high dose temolazide. Had an MRI, and then had the 5 words which every brain cancer patient never wants to hear. Scan consistent with disease progression. And my world fell apart again. It was at that point I was offered an opportunity, and to be honest, I had nothing else, to go on a trial of an immunotherapy, a well-known immunotherapy and a first-in-man immunotherapy. So when I started, these are not the best images to show, but these are the easiest to show because the other ones are quite crappy. This dark spot here sits in my corpus callosum, just above my brainstem, and that was about 136 millimetres squared. Started the immunotherapy. After 4 cycles, it had reduced by 65%. And since then, I've done another 4 cycles and I'm down to 91% regression of that tumour. I only have 2 others and they're tiny. From a year ago, having the avocado, to today, having that spot, which is 3 by 4 millimetres, it's a miracle. And I'm so fortunate. Again, I say all of this is for Luca and Josh. I just want to be their dad.
That's a great job, a really powerful way to showcase what's real about this, what is urgent and what's required. And Misty, if we can throw to you, that immunotherapy piece comes really strong for you. Of course, it's your field, it's your discipline. Can you give us a high-level overview of the immunotherapy system? And then we've got some videos playing in the background. Maybe take us through some of the work that you do in your lab.
Yeah, thanks, Sam, and thanks, Jan, for sharing your really powerful story too. It's really appreciated. We're grateful to have you as a consumer on our lab team and contributing to our science. We all have an immune system, otherwise you wouldn't be here. You'd be in a bubble, not able to participate in the world at all. We all have these white blood cells zooming around our body and our lymph and our tissues all the time. When you get a sore throat and your lymph nodes get all knobbly in your neck, that's your immune system proliferating, dividing and mounting their army. And it's quite possible that many of us in this room have had cancer before and haven't realised it because our immune system has taken it out. So the job of our immune system is to be the sort of surveillance killers, the snipers, if you will, of diseased, affected and infected cells. And so this is what we're showing here. This is a movie here. This is a real live T cell looking down the microscope, swarming along just to show you this is happening in our bodies all the time. So for a lot of us, we can use and manipulate these T cells now in the field, and it's become now the fourth pillar of cancer treatment. So Jan referenced Stupp protocol, so he's had surgery, radiation and chemotherapy, and for a really long time that's all we had. And now we're using the immune system, and we can use a patient's own immune system to either give drugs to ramp it up and wake up a sleepy immune system, rearm it against a tumour, and we can also then genetically engineer it. So we can actually manipulate these immune cells and rearm them in a personalised medicine approach.
Yeah, and we're going to talk about the CAR T you just mentioned. You're going to take us through how that works and what it looks like.
Might stand up if that's okay.
Yeah, absolutely.
This is what a very, very simplified version for what this would look like for a patient if you were to have CAR T therapy for a brain cancer. This has never been done. There's only been a handful of trials on planet Earth, and there have been 0 CAR T clinical trials in Australia. So it's all very new and it's very cutting edge. After diagnosis, the blood would be taken from the patient. The T cells, those white blood cells, would be isolated and grown up and give the right receptor that they need to recognise and kill their cancer, much like a key fits in a lock. It's a very, very highly specialised and specific weapon. They're grown up and then they would be given back to the patient where those T cells, and this is the beauty of the immune system, and you're going to hear from our colleague here, Joe, talk about how hard it is to get drugs across the blood-brain barrier, whereas T cells can cross the blood-brain barrier and get into the brain. So it's they're a really powerful living therapy, if you will.
Yeah, awesome. And we've got an example here of a T cell, I think, delivering its payload. I heard you describe recently.
Exactly. So these are the green cells you can see here are the T cell soldiers, and the blue cells are the cancer cells. And so if you just hit forward again on the cursor there, Sam, on that movie, you can see here that this cell on the right-hand side, all of these red dots are like grenades. They're like the toxic payload that the T cell releases and secretes onto the cancer cell, and it essentially makes the cancer cell blow up. So this is happening all the time in our body, and what we can then do now is develop CAR T cells specifically for brain cancer. So I lead the immunotherapy program here within the centre, and this is the pipeline here that we've developed, is knowing what's on the target, what's different about the brain cancer itself compared to healthy tissue, and how can we differentiate between the two. And we're starting to really get more of a handle on that now. And once we know what those targets are, we can then make these T cell killers, and they're serial killers. So these one T cell can kill many, many targets. And we can have all the development assays that we need to then test how well that those T cells can function through to good models, and then hopefully we can get something into the clinic. That's really the goal here, is to give more personalised medicine treatment options available to patients.
Amazing. Misty, this is a tough one and a big one, but how optimistic are you, or do you get, when you see things working like those videos we just saw? Where does it go from here? What's it done in other cancers, potentially?
I think it's sort of just the hairs on my arm just stand up when I see images like that. It's really exciting. As I said, it's a new field, and immunotherapy, will be a piece of the puzzle in I think new treatments for brain cancer. And this is why we need to work in a collaborative way across the centre. This is the first time we're trying to do team science. So the immunotherapy program sits very closely and works very closely with the brain cancer lab. We work closely with our clinical collaborators. We work with, Andreas Strasser and his team looking at getting some of these new medicines into the brain, and with our colleagues like Joe. So, we all work together to tackle these brain tumours from a multi-pronged approach.
Thank you so much. Really appreciate it, Misty. We're going to, and as I said, we want all of the panel to jump in and out of this discussion. If there's pieces to add, we really want to do that. And also, if there's questions from the audience, please sing out. We do have some that are going to come through from online. And we're going to shift gears now into the clinical world with Kate and Mark, and obviously Jim and Peter. I'd expect that you'd want to jump into this as well. So I appreciate, Kate, again, your attendance, and Mark, I know it's been a big one for you to get to as well. We end up having a discussion here about no progress over 30 years. When we say no progress, how definitive is that? Because it's not as definitive in the surgical or oncology world that potentially people might be aware of.
So I've been doing it for 30 years, thank you.
Yeah,
It's
so
been very successful.
Mark looked like Jim 30 years ago, and Jim will look like Mark in 30 years.
So, Kate and I would say, I'm sure, we have improved the care of our patients and their carers, for that matter, immeasurably. We have done such a good job in improving from what was terrible to what is still a problem. But what we haven't been able to do is make the sorts of changes that we've seen in other cancers. And I've been around long enough to have seen these incredible changes in the care and the outcomes for patients with melanoma, for example, which used to be a cancer that was untreatable. A melanoma that had spread untreatable. We had terrible chemotherapy. We've seen extraordinary changes over the last 10 years or so with immunotherapies. Why hasn't it happened in brain cancer? I don't know. It's a tough cancer. But what I do know, and Misty alluded to it, the team sport. In fact, Joe Biden, when he was Vice President, opened up the VCCC over the road, and he said, cancer research is a team sport. And I think that's really what we've got here. And I was just thinking, Kate and I are probably old enough, along with others, to be the coaches. But gee, what we have done, we have gone out to the draft. Because Kate and I talk about AFL football every Monday morning at length. We've gone out to the draft, and we have found the absolute stars. We have found the top draft picks. And you can see from the people who've talked today, they're all under 40, I think, or certainly under 45 or so. Kate and I are coaching a bit, but these are stars of the future, and they're working together. I think it's incredibly exciting.
Fantastic. Kate, did you want to jump in there about football or otherwise?
So Mark and I never talk about football. I know nothing about football. I'm way too busy to worry about football. I think the really important message is, so over that 30 years, I've gone from a time where, a surgery for a brain tumour, you took a great big bit of the bone off and you basically poked around in the brain until you found the soft bit or the hard bit, and you kind of took that out a little bit, to very targeted surgery, excellent surgical techniques that have meant that patients who once were untreatable in any form can at least have their life prolonged with a moderately good quality of life. We've got better at looking at quality of life. We've got better at managing symptoms like seizures. So there has been progress. What there hasn't been progress in is our ability to then kind of extend that, beyond sort of that the number that everyone quotes as the 14 months or the 5 years for the low-grade glioma, to start really getting runs on the board for a longer survival there. And that's what we really need to work on now. But I think all the work that's gone before of better surgery, better tumour identification, better outcomes from the surgery, better radiotherapy, that really will make a massive difference.
Thanks, Kate. Jim, we talk about this as a team sport. You mentioned it in your talk. We're talking about critical mass and attraction of people. You're attracted to the neurological world. How do we do more of that? How do we entice and engage people?
So, I think from my perspective, let's get this back on. Coming into the brain cancer space was, really clear. The opportunity to have an impact in a field where, although acknowledging the significant improvements in patient care and coordination and multidisciplinary management, the opportunity to be part of the next wave, which is what I think we are currently on. We're just ahead of it. I think of discovery, of progress, of new therapies, I think is an incredibly exciting opportunity for junior researchers to be part of a team. And so what I think we need to, continue to do is to, advocate for this is the place where we want the best scientists and the clinicians and the people to work together and to create the environment that allows people to thrive, to give them an opportunity to fail and to do the difficult science and experiments to actually bring about significant change. And, that's what I feel about being part of this team, to be supported to do difficult things that will take time and to fail, but to hopefully improve and make those significant discoveries in the future.
It's a really important point to balance optimism with time and effort and longitudinal incremental increases in our knowledge and discoveries. Mark, have you got a comment there in terms of how many people we're going to have to bring in over what period of time to make significant change?
Oh, the more the merrier. I think what I've tried to do for people like Jim and others is draw them in to argue that it's a fascinating, from a biological point of view, it's fascinating, but what we aren't doing yet for patients, we might be able to do in the future. And that there's opportunity here. We have attracted some incredibly gifted young investigators, scientists, etc. I think we have got the top draft picks, and there is going to be a burgeoning science and clinical mastery within the field of brain tumours that will just continue to attract and attract and attract.
Yeah. Jim, do you ever think of yourself as a number one draft pick?
No, that's never an analogy that's sort of come to my mind, but I like it and I'm going with it. I think Kate was going to jump in.
Sam, so you've seen Heidi McAlpine, and that fantastic talk she gave. We've got so many talented young people like her who want to go into the field. This is, I think Mark gives a great talk where he talks about when he came back from overseas and nobody wanted to do brain cancer. Now, we've got, so many talented young people who want to either do clinical care or research for brain cancer. What we need to do is give them the opportunity, which means, funding and resources and laboratory space or clinical jobs that allow them to thrive in that, and the mentoring to allow them to reach their potential. So I think we've got plenty of really good people. We need to give them the opportunity.
And final one, Mark, Kate. We've got an engaged audience of 400 or 500 people with a network that's significantly bigger than that, a community of ultra engaged people. What do we wish, as a collective wish that they knew about us, our work, the research, the clinical side of things?
We're trying really hard. Kate and I have been working together for 20 or 30 years. We love what we do. We are desperate to improve the plight of patients and carers, for that matter. We've got an incredible opportunity with the people involved, with the funding that's coming through, with the science that we understand. There's an opportunity here.
Yep. And Kate?
Yeah, I guess, I sit and patients say to me, well, what do we do next, or what else is available? And, I guess I'd like people to know that strikes at our heart just as much when we really don't have anything to offer. Well, not as much as it does theirs, but it really does strike at our heart, and it weighs on our hearts every day. So I want them to know that we're working really hard, and we want them involved. We want them to help guide us, and we want to have a clinical trial available for everyone. So just to let them know that this is what we're working towards, and this is genuinely what we want to provide.
And last one, just because you touched on that there, Kate, the clinical trial platform, Mark, I know you're excited about BPOP, and maybe give us a sense of how long you've been working on that idea, trying to get that funded.
2014. So I would guess 8 years I've been, because we do the same sort of strategy in other cancers, particularly breast cancer and lung cancer and other cancers. I've been watching it done in other cancers, knowing what it could do for brain cancer. And 2014 was the first time I sent an email to someone overseas and said, can't we do this? So it's time to do it, and we're ready to go.
Presented this at a conference sometime around then, and basically everyone just looked at us like we were insane, and said you can't do that in brain cancer. But we've been tacking away for all of those years to say, yes, we can, and this is actually the best way to rapidly gain knowledge about new compounds or new treatments in brain cancer. And, now we're like teetering on the brink of making it a reality. Only a few centres in the world even considering it. So it's fantastic.
And it is a reality. So, in fact, Jim is the chief investigator along with Kate. The study has gone through ethics at Royal Melbourne. It's about to start.
I just wanted to add to that, because when you referenced other cancers, and I sort of came up on that video earlier in the session when I referenced other cancers too, about the level of investment that there's been in other cancers that has been substantial, really substantial. And the area hasn't seen the same for brain cancer. And I think it's why the whole field has stalled so much. We just haven't had the mass capacity. I mean, we need, so this is wonderful, but it is also just a drop in the ocean compared to what it really needs. And, it's been through incredible advocacy to government. It's through people like Liz Dawes. It's through people like Rob DeRose. It's through people that have really, got some shone a spotlight on this, and it's through the hardworking clinicians of people like Mark and Kate that have spent decades telling their patients that they're going to die, and that something needs to change. And so I think this is the start of something, but I think hopefully it can ignite some more excitement so that we can continue to invest in this area, attract the best and the brightest, as Jim points out, and welcome people that want to come and do team science so that we can shift the dial for patients with brain cancer.
Really appreciate that, everyone. Really appreciate that insight into the clinical world. Kate, can you stay with us? Stay with us for a few minutes. We're going to move across to Joe. Joe, one of the 4 discovery themes that we have invested in is the blood-brain barrier. Your work sits within that world. We want you to take us on a bit of a journey of your background and where this blood-brain barrier, which is such a difficult prospect to pass through, makes sense for us to attack.
Yeah, thanks, Sam. So I'm here on behalf of all of these people who form the blood-brain barrier group. And I guess the blood-brain barrier it's really as the name suggests, it is the barrier to getting things into the brain. And the brain wants to protect itself, wants to protect all the delicate cells in there, and so it's built up a really nice system to keep things out. And that's a bit of a problem when we want to try and get drugs in. So if you can go to the next slide, I might and again. So the way I try to explain it is we've got a moat around the castle, and we want to try and get into the brain, and we just can't get across. There's all that water there, but there are ways we can get through. We've got so for example, a bridge here, and we think we're almost there, but there's security guards, and these security guards are just kicking out unwanted people. So what we're trying to do is understand a bit more about these security guards and try and modify the drugs that are being developed that otherwise wouldn't get into the brain, and try and make them look more like guests, so that we're tricking these security guards and we're getting our drugs in there. And so this, within the tumour core itself, within the glioblastoma, this barrier is actually already, it's disturbed. So we could probably get drugs into the tumour core very easily, but the best thing to do is obviously remove the tumour. But there's still a little bit of, I guess, semi-healthy tissue surrounding what's been removed. Can't get absolutely every cancer cell out. And it's in those areas that the blood-brain barrier is more like this and quite difficult to get across. And so even though we get that tumour out, we've got to try get the drugs into the remaining tissue. And that's where our group is hopefully trying to come up with ways to trick the barrier, to try and get our drugs, our antibodies, our proteins, whatever the therapeutic is, into the brain and into the cancer cell.
Thanks, Joe. Are there examples? Are there examples of drugs that can do this, that have been successful in doing this?
Yeah, so one of the most common examples is, it's been around for quite a while, Levodopa, used for Parkinson's disease. So Levodopa is basically replenishing the dopamine in your brain in Parkinson's disease. But if you give dopamine into the bloodstream, it doesn't cross into the barrier. So some clever scientists modified the dopamine to look more like almost an amino acid / peptide, and it gets taken up by a transporter that normally takes the good amino acids in. And when that levodopa gets in, it gets cleaved to dopamine, and hey, presto, you've got your dopamine in the brain.
Joe,
So
I think
it's been going on for
no,
years.
go.
Yeah.
Okay. I think it's incredible because we're all now thinking of our brains as castles, which is cool. But we know that drugs can get there. We know that things like lumbar punctures in surgeries and things like that can work. Why aren't we just doing that?
Yeah, definitely. That's an opportunity to directly inject into the brain or into the spinal cord. But for a disease like cancer, where it's not a once-off dose required, it's something that may require months, sometimes years. We don't want patients to have to come into hospital every week or every day to get that done. So we're working towards trying to get something that's patient-friendly, a tablet, capsule, so that it's can be taken at home as required. Worst case, maybe an injection, like an insulin type thing. But yeah, we try to avoid those sort of more invasive ways.
Joe, really appreciate it. I'm not sure the blood-brain barrier before today for lots of people would have been even a thing or understood. It's, we've got drugs, why can't we use them? It's, as I think Heidi's talk showed, it's an incredibly complex and safe organ developed over millions of years to do what it needs to do to protect us. And one of those things being the blood-brain barrier. So thanks so much and good luck with your work. Peter, we're going to shift over to you in terms of registries and this idea around clinical trials and clinical registries that can support biomarkers. You want to take us through some of your work, and I know Doug touched on this in his talk as well.
Yes, so thanks for the opportunity. Terrific to see all the people here and the engagement. It's just wonderful, I think, for a really difficult brain cancer to treat. So I've done a lot of work over the years in registries. There's lots of registries that have been established for 10 or 20 years in colorectal cancer, in breast cancer, lung cancer, but brain cancer, unlike these, has really been neglected. So there's been no major effort to collect data on brain cancer patients across multiple hospitals. So some have been working very hard, Kate and Mark, collecting data at Royal Melbourne Hospital, but that's just one hospital. We need lots of people involved to make the biggest impact. I think we've lost lots of opportunities over that time to learn from how clinicians treat patients, opportunities for improvement, opportunities for progress. Next slide, thanks. So the Brain Tumour Registry has been made possible by the Brain Cancer Centre. It's a multi-centre effort, so there's now 11 centres participating across Victoria. We're moving interstate, looking to go international. This is a very major effort, unmatched, I think, anywhere else in the world. It's multi-purpose. We're using the data for lots of different reasons. We're not just collecting it for one particular reason, but it's used by many people for many different purposes. And like other registries, I think it's going to have a big impact on how we treat cancer and patient outcomes. Next slide. So just to mention, this is an effort by many people. I'm a clinician. There's many other clinicians involved, but there's research nurses, project officers, data managers, many other people that help in this effort to make it work. So it's comprehensive and inclusive, and just listed a few of the opportunities that the Brain Cancer Registry provides. Better understanding cancer treatment in the real world and how that may compare to clinical trials, some of the things that work and things that don't work. identifying areas of unmet clinical need, evaluating patterns of care, trying to look at ways of improving what we're doing. How do we treat patients at the moment? How can we do better? Particularly exciting, I think, is the opportunity to combine data with tumour samples, looking at better biomarkers, how can we personalise medicine for patients with brain cancer, and also how can we support clinical trials, including clinician-initiated research. So trials are very expensive, often we're limited by resources, often limited by what pharma's interested in. What we as clinicians are interested in may be very different to what pharma's interested in, and the opportunity to leverage the registry data to support trials, I think, is something very important. In the future, lots of different directions we can go in, looking at health economics, helping to assist regulatory decisions, helping to get new drugs available for patients with cancer, helping clinicians to provide the best advice and support for their patient, not just collecting data on the treatment and the outcomes, but also collecting data from the patients, what's their experience in terms of their treatment and the outcomes, and also using the data that we collect to support patient education and decision making. So lots of different ways that the registry data can be used to understand and improve brain cancer treatment.
Peter, thank you so much. Are there any comments either from the panel or from the audience around registries or anything that we've talked about at the moment? How optimistic are we? Who's optimistic? I knew Mark was going to throw his hand up. We're optimistic, right? You know, 2014, you start a conversation that talks about a clinical trial. We're here today. It takes momentum, but...
Yeah, so I said, you know, I used to treat melanoma and I watched it as an untreatable cancer. Untreatable. Terrible chemotherapy. And like that, it became a treatable cancer with patients being cured. There's no reason why it won't happen with brain cancer.
How much, this is a question that's come through online, and I'll throw this to anyone. What is it going to take in terms of dollars to solve this? Over what period of time? Does anyone want to have a crack at that? A lot was Misty's response. Go on, say it, Mark.
100 million?
100 million.
I was going to say
200.
a billion.
How much?
A billion.
A billion.
Right,
But having said that, you know, all of the research I've done over, 25 years of being a neurosurgeon has been cobbled together with, I think my first grant was $5,000 and, bits here and bits there. Over the last 3 years or so, the money that's become available is just incredible. So I don't think a billion is, thinking too much. I mean, I think it'll happen if we start making some gains, people will pour money into it. So I'm really optimistic about that.
So the job now of the Brain Cancer Centre is to invest in research, to communicate results, to continue to drive forward the idea of the Brain Cancer Centre being a collective, collaborative approach, and we move step by step by step towards our vision. And I think that's the path that we go down. I want to thank Jean, Joe, Misty, Mark, Peter, Jim and Kate. Thank you so much for your engagement.